So now let us study about heme catabolism. Heme catabolism mainly occurs in three organs: the reticular endothelial system of spleen, bone marrow or liver, and the liver, and in the intestine. So the hemoglobin is broken into heme and globin, and the globin part is get into the amino acid pool. The heme part is then divided into iron, that is Fe2 plus, carbon monoxide, and biliverdin by microsomal heme oxygenase complex present in the reticuloendothelial system. This microsomal heme oxygenase complex requires NADPH and oxygen, and it is mainly induced by heme. This step is the only step in the body where there is endogenous release of carbon monoxide. Carbon monoxide is mainly required for anti-inflammatory, proliferative, and vasodilation. This Fe2 plus is mainly bound to ferritin, and also that Fe2 plus gets oxidized to Fe3 plus, which is then bound to transferrin. The carbon mono in the reaction of by microsomal heme oxygenase complex, alpha methynyl bridge between first and second tetrapyrrole is broken by heme oxygenase, and this alpha methynyl bridge is liberated as carbon monoxide. Now let us continue the pathway. The biliverdin is then converted to bilirubin. The biliverdin is a linear tetrapyrrole and a green colored compound, which is converted to bilirubin by biliverdin reductase, which is also NADPH dependent. Bilirubin is somewhat reddish yellow in color. Bilirubin can act as is mostly an excretory product and can act as antioxidant. After that, for the transport to the liver, as the liver is the main organ where the catabolism of heme occurs, the bilirubin is transported to the liver by binding with albumin. For one albumin, two bilirubin gets bind on, and it forms unconjugated bilirubin. Why does this unconjugated bilirubin cannot be excreted through urine? This is because it is not water soluble. So the unconjugated bilirubin is transported towards the liver cells, and it is absorbed by the liver by facilitated or simple diffusion. The albumin does not get inside the liver. It gets released, and the bilirubin gets inside the hepatocytes. In the hepatocytes, it is then bound with ligandin and Z protein. This is done to prevent the efflux of bilirubin out of the blood after entering inside the hepatocytes. After that, the main step of the heme catabolism occurs, that is the conjugation of bilirubin. The conjugation of bilirubin is catalyzed by UDP glucuronyl transferase, and it requires UDP glucuronic acid, which is converted to UDP, and it forms bilirubin diglucuronide, which is 80%, and bilirubin monoglucuronide, which is 20%. The main property of the conjugated bilirubin is that it is water soluble. So after that, the water soluble bilirubin is released from the biliary canaliculi through the bile duct as bile acid. This process of excretion of liver is the most important, as it is the rate limiting step of heme catabolism. It is also an active process and is mediated by MOAT, that is multi-specific organic anion transporter, which is also similar to multi-drug resistant protein type 2. And this conjugated bilirubin is transported to intestine. And in this intestine, the conjugated bilirubin is converted to urobilinogen by bacterial enzymes like beta glucuronidase. This is the process of deconjugation. And after that, this urobilinogen is converted to stercobilinogen. That is mostly the fate of urobilinogen is which can be classified into two types. 80% of the urobilinogen gets converted to stercobilinogen, which is excreted as 250 to 300 milligram per deciliter in feces, and it is also responsible for the brown color of the feces. And also 20% of the urobilinogen gets inside the enterohepatic circulation, and out of this 15% gets inside the liver, gets the liver and gets becomes part of the heme catabolism cycle, and the rest 5% mostly gets excreted in the as the urine. So this is all about heme catabolism.